Today we are going to talk about another glycogen metabolism, that is glycogenolysis. So the degradation of the stored glycogen in liver and muscle constitutes the glycogenolysis. So the pathways for the synthesis and degradation of the glycogen are not reversible, and an independent set of enzyme present in the cytosol carry out the glycogenolysis. The glycogen is degraded by breaking alpha 1, 4 and alpha 1, 6 glycosides bonds. So those alpha 1, 6 and alpha 1, 4 glycosidic bond are formed during the glycogenesis. So if you don't know the various steps of glycogenosis, I want you to read the topic glycogenesis, and after you read the topic glycogenesis, I want you to read the glycogenolysis. So as you know that the glycogen is the branch structure. So there are various steps in the glycogenolysis. First I will discuss in brief, and then I will discuss the detailed steps of the glycogenolysis. So first of all, we have the glycogen molecule that have alpha 1, 6 glycosidic bond, and upon action of the enzyme, the glycogen phosphorylase, which is also the rate limiting enzyme. So due to this glycogen phosphorylase, there is debranching of the glycogen and there is the formation of the limit dextrin. Again, again there are various other debranching enzyme like transferase enzymes acts on the limit dextrin, and uh, it further debranches the glycogen, and ultimately the debranching enzyme 1, 6 glycosidase debranches the glucose, and the further action of the phosphorylase forms the glucose 1 phosphate, and on the action of phosphoglucomutase on the glucose 1 phosphate, there is formation of the glucose 6 phosphate, and from this glucose 6 phosphate there can occur glycolysis, or upon the action of glucose 6 phosphatase on the glucose 6 phosphate, there is formation of the glucose. So this is the brief steps of the glycogenolysis. Now I will discuss the detail about the glycogenolysis. First of all, there is action of the phosphorylase. There is action of the glycogen phosphorylase, which is also the rate limiting enzyme. The alpha 1, 4 glycosidic bonds from the non-reducing ends are cleaved sequentially by the enzyme glycogen phosphorylase to yield the glucose 1 phosphate. So during the phosphorylase of dephosphorylation of the glycogen, ultimately there is the formation of the glucose 1 phosphate. So this is also, the process is also called as phosphorylasis. So this phosphorylasis continues until 4 glucose residues remain on the either side of the branching point. Either side of the branching point means that have 1, 6 glycosidic link. The glycogen so formed is known as the limit dextrin. So the limit dextrin, which cannot be further degraded by the phosphorylase, and glycogen phosphorylase possesses a molecule of pyridoxal phosphate, which is covalently bound to the enzyme. So now there is action of the debranching enzyme. There are various action of the transferase activity. So the branches of the glycogen are cleaved by the two enzyme activities present on the single polypeptide called debranching enzyme. So it is known as the bifunctional enzyme. So glycosyl 4 transferase activity removes a fragment of 3 to 4 glucose residues attached to a branch and transfer them to another chain. So here one alpha 1, 4 bond is cleaved, and at the same time 1, 4 bond is made, but the places are different. Amyl 1, 6 glucosidase breaks the alpha 1, 6 bond at the branch with a single glucose residue, and it releases the free glucose. The remaining molecule of the glycogen is again available for the action of the phosphorylase. This remaining glycogen molecule is again further present for the phosphorylase activity. So now there is formation of the glucose 6 phosphate. So the remaining glycogen molecule, upon further action of the phosphorylase, there is the formation of the glucose 1 phosphate. And as I have said earlier, the phosphoglucomutase acts on the glucose 1 phosphate to form the molecule glucose 6 phosphate. So this glucose 6 phosphate, as you already know, is the central molecule that can go to the glycolysis, or upon the action of the glucose 6 phosphatase, it can form the glucose. So this is how glucose is formed from the glycogen, and this whole process is known as the glycogenolysis. I hope you understood this lecture. Thank you.